Being a quick look at cellular respiration and what is happening within the cells, inside those mitochondria to generate all of the ATP that we need for the cells to function normally. So it's within the mitochondria where we're going to produce the ATP. So we're going to start out with carbohydrate, primarily glucose, and that glucose is going to be combined with oxygen to give us energy, as well as combining it with the byproducts as well of carbon dioxide and water. And that's going to allow us to get the energy in the form of ATP. And then we'll have all the energy we need to do our normal processes within the cells themselves. And so with that, we have the ability to run cells as long as we want. The problem is that if we just take that glucose and break it down quickly, we're going to have a huge amount of energy. And so it's an explosive release of energy. This is going to be given off all at one time. So this is similar to the effect of if we were just simply to take sugar and burn it. Light it on fire, whether this is like a piece of bread that catches on fire in your toaster, something like that, or even summertime, so perhaps roasting marshmallows and it eventually ignites. All of that burning that is happening there is the release of energy and heat very, very quickly. We don't want that in the body. We want to take that process and slow it down. We want to take the bonds that are being broken in this process and relatively slowly break them as the process goes along. So instead, we're going to go through this process to generate ATP. We're going to store some of that energy from the glucose molecules and store it in ATP so that we can use it in order to run the systems in our body. So if we are running along, one of the things that we do still get is heat. And so we're still going to have some heat generated. That's going to be good for our bodies because we need to maintain our body temperature. And so heat itself is going to be there to maintain our normal body temperature. If we get too hot, that's where we'll go over to the skin, start sweating, do things to start to cool us off. But if this was done in an uncontrolled process, burning of the sugars very quickly, we would not be able to withstand it, and our body temperature would go up very, very quickly. And so we have to control this process. The way that we control this process is, rather than, in a sense, doing it in just one step, taking our carbohydrate plus oxygen and getting us our CO2 and our water and energy that's there. We want to do this in small steps. So we're going to break down this carbohydrate. We're going to get our ATP from it. And that's going to allow us to do this process very, very slowly. So we're going to relatively quickly break it down. But as we do so, we're going to send it off into little chunks of ATP. This is going to get us our energy release that we need in order to run our normal cellular processes. So for the most part, our cells use glucose as their energy source. So this is our fuel that our cells have for generating ATP. And so this is our fuel, the glucose, but we need to transform the glucose into ATP so that we have little packages of energy. And so these little energy transport molecules that we have are going to transport small portions of energy rather than a large amount of energy. So we're going to go from, once again, our glucose plus our oxygen. And instead of, in a sense, jumping right to the energy, we're going to go through little steps along the way. So we've got lots of things happening in order to get us to that point, and we're going to have small pieces of ATP available for use. And at the same time, it's going to generate a small amount of heat every time that we break those bonds on the ATP. So it's not going to be a huge process from the very beginning. We're going to slow it down and keep those processes running fairly smoothly. So to kind of run through really quickly everything that happens, we're going to have glycolysis. Glycolysis is going to produce 2 ATP. From that glycolysis, we're also going to generate with it some byproducts that we're going to be able to use, namely pyruvic acid. And that pyruvic acid is then going to enter into the mitochondria. Once inside there, it's going to go through the citric acid cycle or the Krebs cycle. As it goes through that Krebs cycle, we're going to get 2 more ATP. And the Krebs cycle is then going to generate hydrogen ions that we're going to be able to utilize for a pump. So we're going to have a proton pump working. Along the way, we've also generated some NADH, some FADH2, and this is going to allow us to also help in this electron transport chain over here and oxidative phosphorylation. And that's going to get us the largest amount of ATP. So we're going to go from this glucose molecule that we started with, and we're going to break it down so that in the end, we're going to end up with somewhere in the neighborhood of 36 ATP. This is where, depending upon how you do the math and which book you're reading, things like that, it can change the total amount of ATP that you have. So anywhere between 32 and 36 is generally what you're going to find in book 4, somewhere there. So essentially we have the process broken down into 2 parts. We have an anaerobic portion and we have an aerobic portion. The anaerobic portion is just glycolysis. Glycolysis is going to happen out here in the cytoplasm. So it's inside the cytosol, inside the fluid that is there. It requires, because it is anaerobic, it requires no oxygen. So we don't need any oxygen for anaerobic cellular respiration to occur. For our aerobic cellular respiration, though, we do need oxygen. We have to have that oxygen there, and so we need to have that available within the cell in order for us to get all of that ATP that we wanted. This process is now going to happen inside our mitochondria. So mitochondria are now going to be used. We're going to take our byproducts of glycolysis. So even if we're doing aerobic cellular respiration, we're still going to jump from glycolysis over to aerobic cellular respiration. And then that's where we're going to go through the citric acid cycle or the Krebs cycle. And then we're going to use our electron transport chain in order to generate all of the ATP that we can. So we get the greatest amount of ATP from that one glucose molecule. If we were to go through and just mix that carbohydrate with oxygen immediately, the energy that would be there would be very explosive, would be very big, but short-lived. So we don't want that. We want to have this energy around and available and in small doses as we go through it. So the first process is always glycolysis. So glycolysis is going to take our 6-carbon glucose and it's going to break it down into 2 3-carbon portions. So we need to have 2 3-carbon portions. Each one of those 3-carbon portions is referred to as pyruvate or pyruvic acid. That's there. And in the breakdown of those bonds, we end up with 2 ATP. So we end up getting 2 ATP out of this first process. If we do not have any oxygen present, so if this one here does not require any oxygen, if we stop right here, we still get 2 ATP and our pyruvate. But the pyruvate portion is going to require us to have oxygen around in order to utilize it. So we have to have that oxygen there to utilize the pyruvate. Otherwise, we could just stop right at this portion here, and we'd have 2 ATP and be done. So from our 1 glucose, we would end up with 2 ATP doing it anaerobically. But we don't want to do that. So we're going to go beyond just glycolysis and allow ourselves to enter into the Krebs cycle or the citric acid cycle. So in order for us to get to the point where we have our 2 ATP and where we have our pyruvic acid, this is where we're going to split our glucose up. And so this requires us to have some enzymes available in order to do this. So we just talked about enzymes, and this is going to catalyze the reaction for us. And as we go through glycolysis, we end up with our processes giving us 4 ATP. But the thing is that in order to get this process going, we had to use 2 ATP. So we made 4, we used 2, and so that's where we end up with only 2 ATP in the end after glycolysis. We still ended up liberating in that process. We still got 2 NADH out of here. And then we also have our pyruvic acid available to go into the Krebs cycle. So it can go over into the citric acid cycle as long as we have oxygen available. If there's no oxygen available, we're going to move over, and that pyruvic acid is going to get converted into lactic acid. And that lactic acid is what's then going to start to build up in those cells. This is where, if you've ever used a muscle over and over and over again, you're working out, things like that, as that muscle begins to start to fatigue and it loses its available oxygen, lactic acid starts to build up inside that muscle. And that's the end of the story, because now instead of getting 32 to 36 ATP out of that one glucose molecule, we only ended up with 2. So this is where we want to be able to go from glycolysis. We got our pyruvate ready. Now we can take that pyruvate and put it into the citric acid cycle. So the citric acid cycle is then going to convert that pyruvate over into acetyl-CoA. This acetyl-CoA is now going to allow us to take our 3 carbon pieces that we have and break them down even further. So we're breaking bonds between carbons along the way. So this is where we're getting that energy from in order to liberate it onto our ATP. This is now happening once again inside the mitochondria, and it requires oxygen. We have to have oxygen present in order to do this process. As we are breaking bonds on the oxygen, we have our hydrogen bonds that are there. Those hydrogen bonds keep on getting broken off, and then they're picked up by hydrogen carriers. In this case, our hydrogen carrier is either NAD plus or FAD. And so we take NAD plus, we're going to add our hydrogen, and we end up with NADH. And then our FAD is going to pick up 2 hydrogens, and then we end up with FADH2. In this process, we now picked up 2 ATP. And so 2 more ATP are going to be produced during this process. Since we've taken the hydrogen off of the carbon molecules themselves, we are now left with some waste product. We now have carbon dioxide. So that Krebs cycle is a relatively complex series of chemical reactions. And so our pyruvate enters into the citric acid cycle, or the Krebs cycle, and it's going to go around this whole process here and all the way back to the beginning. One time around, we make ATP. So that means that we have to take that pyruvate around twice. We're going to have 2 pyruvates that we had. We're going to take that around twice, and that's where we get our 2 ATP from. So we end up with our 2 ATP because we go around there 2 times. Here's where we created our acetyl-CoA. And so we have our acetyl-CoA being generated within that process, and we start now to go through the entire process. We can see that we have our NADH being developed, we have our FADH2, and we're ready to move on to the next step. Now that we have our hydrogen carriers, we have a way to hold on to that hydrogen inside there. We now have a process that we can continue to move on from at this point. So we've made it from glycolysis. We made 2 ATP there. We made it through the Krebs cycle, or the citric acid cycle. We made 2 more ATP there. But if we're going to end up with 36 ATP total, that means that 36 ATP's got to come from somewhere. So we're short, we're at 34 ATP. We're going to need at least 32 more. That's where, now that we have something that is holding onto our hydrogens, we're going to be able to use those hydrogens in order to, as a pump. And so this is all going to occur now in the electron transport chain and oxidative phosphorylation. So the last step itself is our electron transport chain. This process is going to take our NADH and our FADH2 that we got from the citric acid cycle, and this is going to enter into this transport process. And so essentially we're going to move hydrogen ions through a membrane. And as we move them through that membrane, that's taking energy to do so. So we have a high concentration. So our hydrogen ion concentration is high, and that's going to drive the process of moving those hydrogen ions through. And when that happens, that's effectively going to take kind of what amounts to a little protein here, and it's going to spin it around. And as it does so, that's going to allow us to generate our ATP. And so electrons from the hydrogen are passed through that protein here. And so as the hydrogen goes through that protein, essentially an enzyme that is there, that's going to allow us to produce ATP. And now our ATP is going to be produced at this point, and it's going to run and run and run and run, and we're going to get somewhere in the neighborhood of 30 to 34 more molecules of ATP, plus some water this time. And so now we have some byproduct of water coming through the process itself one more time. And so here we have that, the proton pumps themselves. And so here we have the pump. Our hydrogen ions are going to be able to come through that pump. As they come through that pump, they're then going to come over here to this enzyme. And as they do so, that's going to effectively kind of rotate around. And when that does so, ADP is then going to be combined with inorganic phosphate. And so inorganic phosphate is now going to be added to that ADP, and we end up with our ATP here. So we need an enzyme. We said before that enzymes end in ASE, so we have ATP synthase. Now that we have all of this hydrogen on the outside, our enzyme can then take the energy that is stored by having a high concentration of hydrogen ions out here. That energy can then be taken through. And so this is like similar to when we talked about having a concentration gradient, where we had our kinetic energy that was there. We have this movement that is going through there because we created a potential energy in the first place. So we have a hydrogen ion concentration, but as we move those hydrogen ions through, so our concentration is our potential energy, the kinetic energy is the actual movement of that hydrogen ion, and that then, erase that a little bit, that then moves our ATP synthase, and that combines together our ADP with our inorganic phosphate, and we now have all of that ATP that we need. So sum it up, we have our 2 ATP coming from glycolysis. We can stop there, be done. Instead of getting pyruvic acid to go into the mitochondria, that pyruvic acid can then be left outside here, and we're going to have a buildup eventually of lactic acid as it gets converted into it. Or we continue on, and that pyruvic acid comes and enters into the Krebs cycle. From the pyruvic acid, we convert it into acetyl-CoA. We get 2 of those from our 2 pyruvates. That's there, come into the Krebs cycle or the citric acid cycle, go around that twice, generate 2 more ATP. We now have all of our hydrogen carriers, and those can then go into our electron transport chain, move through and across the membrane, and as we do so, we're going to generate our ATP. And this continues on until all of those hydrogen ions have been used up. So we're going to need, in this process, oxygen. We have to have that oxygen there for this final step. If we don't have it there, we're going to have to be accepting of the fact that we're only going to be able to do anaerobic respiration. So once again, anaerobic respiration, no oxygen. We don't need any oxygen for this process. In our own bodies, we do lactic acid fermentation. And so we're going to generate lactic acid. But if we were to give that pyruvic acid to, say, yeast, and so as we break down sugars in something like wine or beer or alcohol in the process itself of generating grain alcohol, the yeast that is inside there generates alcohol instead of lactic acid. And so other organisms like yeast or bacteria, if we want to make like yogurt, things like that, they do the same thing, and they're going to go through this process that's there, and they're going to stop there. So this whole process here of glycolysis, our glucose comes in, we generate 2 pyruvate, we generate 2 ATP, and then we stop. We're no longer going to go from this process, and we're going to end up with 2 lactates, 2 lactic acids, and this is going to create that burning sensation in our muscles if we're using our muscles in this process itself. So we don't want to use anaerobic respiration if we can help it. We want to typically stay in aerobic cellular respiration. And we'll get into that more when we start to talk about muscle contraction later on.